Ladies and gentlemen, it gives me great pleasure to talk to you on this interesting and emerging topic called the diabetes, obesity, and the brain. What I want to do is to run through the topic with a fresh review with lots of data emerging. And if you see here, the American Heart Association's slide from 2017, where they conceptualized and looked at brain health and cognitive dysfunction as a primarily as a vascular problem. So we know this macrovascular and microvascular complications as well as cardiovascular diseases like atrial fibrillation and arrhythmias and cardiomyopathy that gives rise to vascular issues to develop cognitive dysfunction. And on the other hand, they also nicely tell us about the genetic components, the neurodevelopment, the social issues, and other behavioral issues like diabetes and obesity that can give rise to either optimal or suboptimal or deranged brain health. In the same paper, they also talk about Life Simple 7, where they talk about primary prevention of cerebral vascular disease leading to brain health deterioration. So they have given 4 ideal health behaviors and 3 ideal health factors. So as you can see, the health behaviors for primary prevention of brain dysfunction or cognitive decline is non-smoking, increasing physical activity, consuming a healthy diet, BMI in the non-obese range of less than 25 for Americans. And then the 3 ideal health factors include a normal blood pressure less than 120/80, a cholesterol that is well within the normal range less than 200, and a fasting plasma glucose that is not deranged and it's less than 100. So these are generally considered to be the vascular disease prevention risk factors that will give rise to or give rise to protection from cognitive decline at old age. However, what I've done for this paper is to analyze PubMed with the words obesity, diabetes, and brain. And as you can see, I turned up with more than 800 papers, and I've looked at the last 2 years' papers in particular, and then I have followed back to see what the literature says. And what I found and what I want to share with you today is to talk about optimal brain health or brain health as a function that is not just vascular, but a much broader sphere. And we'll talk to that as of the perspective of an endocrinologist from a molecular and behavioral point of view. So you can see the multiple factors that either optimize or derange or impact brain health include the gut-brain axis, which I will speak about, the neurovascular unit, central insulin resistance, which is a new emerging concept, and then we'll talk about the impact on cognition from all of these factors, and inflammation, which is inflammation leading to aging in the brain. So I'll take you through each of these concepts and then I'll conclude. So first of all, the neurovascular unit and the blood-brain barrier is something that we have come to know of. So if you look at the capillary, it has this basal lamina, which these tight junctions, which does not allow substances to leak into the brain. So that gives protection to the brain. However, this blood-brain barrier permeability is an important concept in inflammation and in certain toxins and certain diseases. We know that this basement membrane can be damaged. And we also now have emerging evidence that in insulin resistance and with various other factors, this can get deranged, which is what I will talk about. So the models that people have used to study cognition is the zebrafish. You might say, why use fish brain? The zebrafish has a particular characteristic of the brain in that as compared to human brain, which cannot regenerate, the zebrafish brain is far superior to our own brain because it can regenerate. So you can do studies with models where the brain is deranged or inflamed, and then whether it can regenerate and so on. You can also feed the fish with various types of diets to see what kind of impact it has on the brain cognition or on the biopsy features of the brain. So if you give high fat diet, for example, or high carbohydrate diet, it has been shown that the brain of the zebrafish does get damaged. And as you can see here, it leads to blood-brain barrier leakage, leading to the stem cells, which are important for the zebrafish to regenerate its own brain, become under tension. So oxidative stress as well as inflammation occurs, and these factors that derange it include various dietary patterns and giving it high fat and high calorie diet. So as you can see, these are the various factors that lead to the problems in the zebrafish: hyperglycemia, insulin resistance, adiposity, hyperlipidemia, liver steatosis, which we will speak about as an important predictor of brain health today, inflammation, which I will keep going on and on about, oxidative stress, hormonal disruption as well, which we will speak about. We also know that these are the problems that occurs: blood-brain barrier dysfunction, inflammation, the neuroinflammation, we call it, oxidative stress, decreased brain plasticity. And in zebrafish, we also know that this neurogenesis and regeneration is impacted by noxious stimuli. As we've seen here, the stem cells are important for regeneration, and they come under attack when there is inflammation and various other damages like steroids, oxidative stress due to various dietary factors, and diabetes, etc. We also keep talking about this tau protein and beta amyloid protein, which are shown in these, the beta amyloid and the tau, which accumulate in the brain and which is seen even in humans as a marker of neuroinflammation and neurodegeneration. And then we go on to talk about the next topic, which is the gut-brain axis. So to put it simply, you can see that the brain is innervated by the vagus nerve, which senses and takes signals back from the gut and also gives signal to the gut. And in the gut, you see these lining epithelial cells and the enteroendocrine cells, which are hormone producing, which are lining the tissues. And then you have microbiome, which is an important concept for health. And so microbiome are the digestive assistants to the body. There are trillions of them in our cells and inside our gut, and they interact with the gut in order to bring about health or disease. So if you see the enteroendocrine cells, we know the satiation hormone ghrelin that comes from the stomach induces appetite. And on the other hand, in the intestine, as the food goes from the stomach into the intestine, the stomach produces several hormones like cholecystokinin and GLP-1 and PYY and GIP. These four are important satiation signals which talk back to the brain. If you have absence of enteroendocrine cells, for example, in this kind of patients who have been reported to have neurogenin 3 mutation, which leads to absence of enteroendocrine cells in the gut lining, they don't have the ability to produce these enzymes for digestion, and they have malabsorptive diarrhea. So the endocrine system in the gut becomes a very important concept in digestion and weight control, obesity control, etc. And in this cartoon, you can see further details of how this works. The gut microbiome interacts with the gut in multiple ways, as I will keep expanding this concept of gut microbiome and gut health and brain health. And the enteroendocrine cells, which respond to the nutrients entry, which is usually modified by the gut microbiome because they are our digestive assistants, and they themselves sometimes influence these signals. and then hormones are produced from these enteroendocrine cells, which then go back to feedback on the brain. And here we can see how entry of nutrition changes the secretion of hormones. And we also know that in the brain, there are certain types of cells in the appetite center, in the arcuate nucleus of the hypothalamus, that increase appetite as well as decrease appetite. So the top ones increase appetite, and the bottom ones, like the GLP receptor, actually decrease the feeding and create a satiation response. So as I said, there is this brain center in the hypothalamus, which reduces appetite and increases energy expenditure in a fed stage, leptin, and also GLP and somatostatin and CCK, cholecystokinin, give that response back to satiation and increasing energy expenditure, whereas ghrelin, when in a hungry stage, when the stomach is empty, gives rise to the signaling to eat and desire to eat. So we can manipulate this gut-brain axis to bring about weight changes. For example, this is a history of the weight reducing drug development. As you can see, most of them influencing the appetite center in order to bring about, or also influencing the adrenogenic, the receptors and the neurosympathetic signaling to produce more energy expenditure are the drugs that have been there. As you can see, some of these are available still currently in use, include phentermine, orlistat, topiramate, phentermine combination, naltrexone, bupropion combination, and then GLP-1 analogs, whereas lots of the others have been discontinued due to adverse effects. Now, talking about the GLP-1 receptor agonist, which I will expand in a few slides, you can see that although there is a blood-brain barrier, some of these molecules like lixisenatide and the smaller molecules like exendin and exenatide can cross the blood-brain barrier and enter the brain. Larger molecules like liraglutide cannot actively diffuse, but are actively pumped into the system and can reach the brain. And GLP-1 receptor agonist like exenatide or liraglutide can activate the AKT signaling system in the brain to bring about regeneration or stopping the oxidative damage to the brain and offer neuroprotection, an exciting new area where we, particularly in diabetes and obesity, where these drugs are used, that they are not just produce weight loss by satiation signals, but also have anti-inflammatory effect within the brain to give neuroprotection. So it's an exciting area to see where this would lead to in terms of treatment of neurodegeneration. We also know that diabetes is a particular risk factor for Parkinson's disease. As you can see in this UK cohort of more than 8 million patients, 2 million of whom had diabetes, the hazard ratio for new Parkinson's disease was 1.32, telling us that diabetes is actually a major contributor or a risk factor for development of Parkinson's. Now, GLP-1 receptor agonists can restore dopamine neurons in the Parkinson's disease model in animal models. So here is the first one, what is labeled as D is the MPTP compound which causes Parkinson's without dopamine in the brain, MPTP damage, but liraglutide added a GLP receptor agonist. You can see the dopamine and also similar signaling, but much more remarkable one for semaglutide, which is another GLP-1 receptor agonist. So ladies and gentlemen, it's exciting to see that diabetes therapies can actually bring about, through the GLP receptor signaling, brain protection and even restoration of the dopamine in the brain. We spoke a little bit about gut microbiota and what to do with them. Now, lots of studies are looking at this early stage of experimentation and research into what kind of food could give you a healthy microbiota. So a wide variety of vegetable, fruits, whole grains, plant-based protein, and fermented foods can give rise to types of live bacteria that are associated with healthy microbiome in the gut. These gut microbiota are not just digestive; they sometimes break down and sometimes produce neuroactive products like dopamine signaling, sometimes noradrenaline-like molecules that then can potentially influence the enteroendocrine cells to affect the gut hormone production. This research is still in early days, but it is very exciting because the bacteria that we thought were harmless commensals in the gut are themselves responsible for occurrence of various signalings within the body and can modify and are associated with obesity and diabetes occurrence. So we know that these enteroendocrine produce GLP and PYY and all that gives rise to the satiation signals in the brain, as opposed to healthy diet patterns. Unhealthy, highly processed and sugary and high fat diets can give rise to and are associated with altered gut microbiota, which can then the types of bacteria that overcolonize the gut can give rise to abnormal signaling and can also lead to breakage of the gut barrier and take away the mucosal protection, leading to invasion into the cells, creating inflammation. The broken down lipopolysaccharides and other molecules that these gut microbiota produce can cause systemic inflammatory response. And we are now studying whether the leakage of this is associated with neurodegeneration and also whether change in the diet patterns and lifestyle and avoidance of toxic behaviors like alcohol and smoking could give rise to a regeneration of the gut microbiota. And there are lots of new studies that are looking at this in terms of brain health, anxiety disorders, and sometimes depression, neurodegeneration, Alzheimer's are all associated with dysbiosis in the gut. And as you can see, this what is called dysbiosis, abnormal proliferation of certain types of bacteria in the which being predominant unhealthy types of bacteria can give rise to various kinds of disorders, including brain disorders and neuroinflammation. The good news is that in experimental models, this inflammaging, which is the inflammation leading to deterioration. For example, these animal models show us the concentration of the A-beta amyloid deposition. You can see it's highest in the model where the abnormal dysbiosis of the bacteria occurs. And certain types of bacterial probiotics like bifidobacterium or introduction of AKT, as I've shown you, is a neuroprotective mechanism within the brain that induces lack of or protects against inflammation, can all give rise to decrease in the A-beta amyloid plaque accumulation. And therefore, this is an exciting new area which we are carefully watching. These are still animal models, but as you can see, even the gut-brain axis is largely responsible for how the brain responds to certain signals in our environment. I also want to talk about exocrines. When exercise is induced, a whole lot of beneficial changes occur in the brain, like reduction of abnormal signaling and increase in things like adiponectin, which are beneficial to the metabolism. And so if you look at exercise and adiponectin, you can see that these beneficial chemicals crossing into the brain sometimes and increasing the brain function, neurogenesis, and improving cognition and mood, and increasing synaptic plasticity, which can lead to beneficial effects for the brain. Moving on to my third topic, which is central insulin resistance. We always talk about peripheral hepatic and musculoskeletal resistance, but even in the brain, in the neural tissue, you can have insulin resistance as a result of inflammation and peripheral adiposity, which as in health, this blood-brain barrier is intact and the neural tissue is fine. And when there is increased inflammation and inflammatory molecules entering with low adiponectin, then you can have reactive microglia, which are inflamed and that can itself induce insulin resistance, reduces satiation, and increases the sympathetic nervous system outflow to adipose tissue, leading to further increase in adiposity. So you can see that insulin resistance can induce adiposity and increase in satiation. So in this experimental model, you can see the normal tissue in the hypothalamic arcuate nucleus. Normal signaling occurs, AKT working normally, which results in food intake being reduced or satiation occurring, and then lipolysis and brown adipose tissue thermogenesis happens, which is beneficial, and white adipose tissue becomes brown, and the brown is the better fat in the body, as you know. Whereas when there is inflammation and resulting insulin resistance in the hypothalamic tissue, this signaling is deranged. There is no more AKT and inflammatory molecules increase, which leads to increasing food intake or satiation, lack of satiation, the decrease in the thermogenesis, and decreasing in browning of adipose tissue, which leads to yellow or white fat, which is increasingly being deposited in the body. So insulin resistance itself can give rise to further adiposity, creating a vicious cycle. But you can also see here how insulin resistance can increase A-beta amyloid deposition and also further inflammation through the mTOR pathway, all of which give rise to degradation of the brain tissue and can give rise and can potentially be risk factor for Alzheimer's and other types of vascular dementias. Another thing that we spoke about at the early on in the lecture is the fact that non-alcoholic fatty liver disease, once again, an inflammatory condition associated with insulin resistance and gut dysbiosis, can be associated with hyperammonemia, partly because of the gut dysbiosis, partly because of the liver dysfunction, and can give rise and be associated with insulin resistance and can itself be neuroinflammatory because of its various associations, and the further hyperammonemia can give rise to neuroinflammation and be toxic to the brain. So, ladies and gentlemen, we looked at various concepts of new outlook to the brain health, as I've shown here, the gut-brain axis being an important determinant of brain health. Neurovascular unit as a protective mechanism and how it can be deranged in inflammation and dysbiosis of the gut. We spoke about central insulin resistance and how adiposity can become a vicious cycle. We spoke about how cognition can be affected by deposition of A-beta amyloid and the tau protein, and also by inflammation, because not all the patients with Alzheimer's have excessive plaque deposition, but in neuroinflammation can be silent without morphological features. And we spoke a lot about inflammaging in this lecture on how inflammation leads to brain degeneration. We also spoke about how we have tried to interfere with this gut-brain axis in a positive way to bring about weight loss and the resulting many molecules that are in use. And we spoke about the new pathways in which research is going about GLP-1 receptor agonist and neuroprotection in terms of decreasing brain cognition decline, and also dysbiosis and neuroinflammation, how gut microbiota can be deranged, and molecules like indole and TMAO can give rise to not just brain dysfunction, but increasing atherogenic cardiovascular disease, and they can also be associated with generalized inflammation and various other disorders. And we looked at briefly how in experimental models this has been used to cut down the A beta plaque accumulation and leakage of lipopolysaccharide to bring about brain health by changing the gut microbiota to a healthy one. Should we teach our patients to eat well for the brain? This is something that we need to think about. A lot more research is needed as to how to change the gut microbiota to a healthy colony and to be less harmful to the body. And we also saw how insulin resistance in the brain can give rise to damage to the tissues, can give rise to tau and A beta amyloid deposition, and how insulin resistance can be a vicious cycle and can give rise to further adiposity, further enhancing Alzheimer's. We spoke about exocrines like adiponectin, which can be protective to the brain. And I hope I left you with some positive impacts about how we can change our patient's life by looking at simple measures like exercise and the type of diet they eat, and a better understanding of the gut-brain axis, and a better understanding of hormones and how insulin resistance impacts patients will either determine health or deterioration of cognition. And I thank you for giving me this opportunity, and thank you for watching.